Hi everyone, welcome back. This week we're talking about cells and we're going to be covering cell structure and function and membranes and cell transport. We have a lot to cover in this lecture, so let's get started. So the learning objectives are to be able to describe what a cell is and the two general types of cells, describe the structure and function of cell membranes, describe several ways in which molecules move across membranes, describe how cells are connected and how they communicate with each other, and be able to describe the 9 important landmarks in a eukaryotic cell. So that's our objective. A lot to learn. So here we have a photomicrograph, an electron scanning micrograph of a cell. Extremely beautiful. Nature is really beautiful in its architecture. But here we can see all the different organelles that are packed within the cell. But let's start with our intention as to why this is even important. Why do you need to learn about cells, right? Because everything that makes up you is all about a mass of cells. You are a mass of cells. And it's great when they're all working together in concert as a team to keep you healthy and to keep you functioning. But when cells go wrong, when something goes wrong with the cell membrane, when something goes wrong with the DNA, when something goes wrong with the transport that we're going to talk about, then that's when we get disease. So it's important to understand the cell on its very basic level because you're understanding how disease happens and how you function, okay? So all organisms are made of cells. And the cell, this is definition, you're going to have a lot of terminology in this lecture. The cell is the smallest unit of life that can function independently and perform all necessary functions of life, including reproducing itself. And with that definition, we can see the huge diversity of organisms that we have on this planet. So here we have this beautiful pixie parasol mushroom, this ring-tailed lemur, this disgusting head louse. All organisms are made of cells. So here's a theory that you need to know. Everyone who takes biology needs to know and understand this. So this is the cell theory. So the cell theory is one of the unifying theories in biology and one that is universally accepted by all biologists. And the theory states that, number one, all living organisms are made up of one or more cells. And number two, all cells arise from other pre-existing cells. Okay, they don't just magically appear. So that is cell theory. And we can see that you don't have to actually work in a lab and use a microscope to see cells in action, right? You could open your refrigerator and you probably have eggs in your refrigerator. That's a cell, right? And it turns out cells are actually one of the most valuable objects on the planet. So when we look here at an egg, which is a cell, it doesn't have to be really small, right? We can see this ostrich egg, which weighs more than 3 pounds each. That's a huge cell. All the way down to this beluga sturgeon caviar. These are fish eggs, and they can sell for more than $700 an ounce. And this is what a sturgeon looks like that makes these caviar eggs. But the most expensive cell on the planet is actually the human egg. And so human eggs can fetch as much as $25,000 for a dozen eggs, as compared to human sperm cells, which can command a few pennies for $20,000 cells, because they're very hard to obtain, right? Eggs are, human eggs. So when we look at cells, we actually break them into two basic categories that you need to know. One is prokaryotic cells, and the other category is eukaryotic cells. Now, when you think prokaryotic cells, I want you to think of bacteria, because they are one of the largest components of the prokaryotes. And the major difference that I want you to keep in your mind between a prokaryotic cell and a eukaryotic cell is that a prokaryotic cell does not have a nucleus. Its DNA simply resides in the middle of the cell, okay, attached to the cell membrane. So prokaryotic cells do not have a nucleus. On the other hand, eukaryotic cells, of which all of our cells are eukaryotic cells, we are eukaryotes. Eukaryotic cells have a central control structure called a nucleus, which contains the cell's DNA. So if I show you a picture on an exam of a cell and you see a nucleus, and I ask you, is this cell a prokaryote or a eukaryotic cell? You see a nucleus, you automatically know it's a eukaryotic cell, okay? So that makes it very easy to determine those two categories. Prokaryotes, no nucleus. DNA is just free within the cell itself. Eukaryote has a nucleus. We are eukaryotes, all right? So science classifies things. We like to keep order, right? And we look here and we can see that prokaryotes are all single-celled organisms, and bacteria is part of that huge classification. And then you have this line of demarcation, and I'm going to try to ping my cursor here so you can see where I am looking at the image. So prokaryotes are bacteria, and this other brand new classification that was recently found, recent in mankind terms, called the archaea. In the archaea, they're prokaryotes, but they're the extremophiles. They're organisms that live in the extremes on the planet. So places that we previously thought no organism could ever exist. So in acid pools, in certain regions of the world, we thought nothing could live in that. We find bacteria that are classified now as archaea. When you go down to the depths of the ocean, there are these volcanic vents where extremely high temperatures are. I'm going to show you an image in a bit, where we previously thought nothing could live close to those vents. Lots of life is flourishing in those extreme temperatures. And the organisms that live in those extreme temperatures, they're part of the archaea, and they are prokaryotes. On the other side of this dividing line of classification are the eukaryotes, and that's us and the other animals that you're very used to on the planet, like us and dogs and cats. But here, when we take a look at the archaea, we can see prokaryotes. We can see that here is an image of a volcanic vent at about 400 degrees centigrade. Now, I want you to think about that when we talk about centigrade versus Fahrenheit. Centigrade is very easy to think about. So 0 degrees centigrade is freezing. Water freezes at 0 degrees. 100 degrees centigrade, water boils. So centigrade is really easy to think about, right? So when you think about that, water boils at 100 degrees centigrade. And here the temperatures at some of these volcanic vents are 400 degrees centigrade. That's tremendous. But yet we find all of this diversity of life. I'm pinging my cursor here for you. So this subsea floor thermophile was isolated from this deep sea hydrothermal vent, and it it actually eats sulfur and hydrogen and fixes its own carbon from carbon dioxide. So it uses sulfur as fuel, which is, again, we thought was something that could not possibly happen. But yet, we have found these extremophiles, and they have their own classification in the prokaryotes as part of Archaea. Okay? So let's take a look at basic prokaryotic structure. Remember when we were talking prokaryotes, I want you to think bacteria. That's a really easy way to kind of keep those classifications straight in your head. So we can see that the DNA is free. There is no nucleus. That's what makes a prokaryote. But they do have a plasma membrane that's containing the structure of the cell. They have cytoplasm, this jelly-like material on the inside. And the way to think of cytoplasm, I think the easiest way to think of cytoplasm is like when you go to your refrigerator and you have an egg, right? You have the shell on the outside of the egg, right? And there's this other really thick white membrane, like when you're peeling an egg, there's this thick white membrane. Think of that as the plasma membrane, okay? And then think of the egg white as the cytoplasm, because that's basically a good correlation for thinking about what cells actually look like, because an egg is a cell, right? And then the yolk, in your mind, think of it as the nucleus, right? There is another nucleus that's in there in that yolk, but on the big picture, just trying to correlate and make sense with what you already know, I want you to think of it that way. So it kind of makes sense, right? And we know that prokaryotes do not have a nucleus, that their DNA is free within the cytoplasm, okay? And then here we can see these little dots, which are ribosomes. And I am going to try to zoom in a little bit so you can get a closer look. Hopefully I won't completely break everything here. There we go. We can see these little dots. These are ribosomes. And ribosomes you're going to learn more about. They're actually these granular bodies in the cytoplasm of prokaryotes. We also have them in eukaryotes. But they're in the cytoplasm of prokaryotes, and they convert the genetic information that comes from the DNA into proteins, into all the different proteins that the cell needs. Now, prokaryotes can sometimes have cell walls. We like to think of cell walls as being only in plants, but you can have prokaryotes having cell walls. They can have what's called a capsule, which is its protective outer coating, right? So cell membrane is here, and then above that, you could have a capsule. Not all of them have a capsule. These are additional structures that they may or may not have, depending on the organism itself. They all have these four structures that you see here: the plasma membrane, the cytoplasm, DNA, and ribosomes. All prokaryotes have that. Now, some prokaryotes could have a cell wall. Some prokaryotes could have a capsule. Some prokaryotes could have what's called pili, and these are like hair-like projections that help the cells attach to other surfaces and sometimes play a role in DNA transfer. And some prokaryotes can have flagellum, and that helps them to actually become motile. Okay, not all. So there's a difference in the additional structures that they may have. Okay, but they'll all have these four basic structures: plasma membrane, cytoplasm, DNA, and ribosomes. All right, let's carry on. But when we look at eukaryotes, the cells that actually make up us, let's take a little step back and take a look at another slide. So eukaryotic cells have these compartments with specialized function, and these compartments are called organelles. We're going to learn more about the organelles, and you need to know the organelles, okay? But eukaryotic cells, plants are eukaryotic cells. Of course, mammals, all the animals on the planet, they're eukaryotic. We are eukaryotic. Slime molds are eukaryotes. Who knew, right? But eukaryotes all have organelles, right? And so now we can compare and contrast eukaryotic cells versus prokaryotic cells. So, major difference, eukaryotic cells have a nucleus, and that's where the DNA resides. Prokaryotic cells, no nucleus, okay? That is the big picture I want you to remember. Now, a lot of people tend to get confused and not want to put plant cells into the eukaryotic category, but they are eukaryotes. But I want to take a look at and contrast the animal cell versus the plant cell in its basic structure. So, two things that I want you to remember that plant cells have that are different than animal cells. Number one, plant cells will have a cell wall that gives them the structure, that gives them the rigidity. Animal cells do not have cell walls. We don't have cell walls. We have plasma membranes, okay? Plant cells have cell walls. They also will have plasma membranes, okay? Plant cells will have chloroplasts, and that's where they're actually conducting photosynthesis. We're going to learn more about that as we move forward. Animal cells do not have chloroplasts. We cannot conduct photosynthesis. We can't use light to actually make energy the way plants can. Now, here's a point that most people get confused on. Both plant and animal cells have mitochondria. These are the little batteries, the little powerhouses of a cell. And plants have them too. So plants have both mitochondria and chloroplasts in addition to cell walls. And then they will have all the other organelles that we're going to talk about in animal cells. So if on an exam I show you an image of two cells and I ask you if, say, the cell here where my cursor is a plant cell, and I clearly label that this is a cell wall, and I ask you, is it a plant or an animal cell? Because there's a cell wall, you instantly know this is a plant cell. Animal cells do not have cell walls. You also instantly know that this is a eukaryotic cell. Why? Because there's a nucleus, right? Okay? So make sure you understand those differences. So, here's something very interesting. Humans, deep down, may actually be part bacteria. How can that be, right? Well, there's a theory called the endosymbiosis theory, which I need you to know. And this theory actually has been developed to try to explain how eukaryotic cells became so complex, how they actually developed all these different organelles. So the endosymbiosis theory was developed to explain the presence of two organelles in eukaryotes in particular. Number one, the presence of chloroplasts, and that's in plants and algae, and the presence of mitochondria in both plants and animals. And the reason why those two organelles in particular needed some form of an explanation as to why they exist, it turns out that I'm sure you've learned that our DNA is in the nucleus, right? 23andMe, we'll talk more about DNA later. Your DNA is in your cell's nucleus. Well, it turns out that the mitochondria that are in your cells, which are the powerhouse of your cell, they make the ATP, the energy that you need, they have their own DNA. And it's different than the DNA that's in your cell's nucleus. Okay? And everyone's mitochondrial DNA comes from their mother. Okay? So it's called maternal mitochondrial DNA. So why would we have an organelle in our cells that has a different set of DNA? So let's take a look at the endosymbiosis theory that tries to explain this, the existence of mitochondria and chloroplasts. Okay. So one thought in this theory is that here we had this ancestral cell to our eukaryotic cells, right? Way, way back when. And then we had this ancestral prokaryotic cell. It was a prokaryote that was trying to live all by itself. And its whole job was that it was really proficient at converting food and oxygen into energy. And at some point, it is thought that the eukaryotic cell engulfed the prokaryotic cell. And this ended up being this perfect relationship where you had this cell that wasn't so good at making its own energy, but once it engulfed this prokaryotic cell that was fantastic at making energy, it was the perfect combination. And over time, the engulfed prokaryote evolved into this organelle that we now call the mitochondria, or in the case of plants, to explain the existence of their chloroplasts. Okay. The endosymbiosis theory goes on to explain some other organelles, which you will be learning about very soon in coming slides, that have sort of this folding nature to them. They look like pleated sheets, right? And so the thought is, again, way back when we basically had just these cells, right, that at some point the plasma membrane started to fold in on itself, and then we ended up with these inner compartments, which are now organelles being formed, and they ended up becoming what is now what's called the rough endoplasmic reticulum. So trying to explain the existence of this sort of folded material that is the rough endoplasmic reticulum inside the cell. Okay? So organelles may have developed by endosymbiosis or invagination, or a combination of the two is the endosymbiosis theory. Okay? But I want to carry on and look at cell membranes, because cell membranes are basically the gatekeepers for the cell. Okay? And remember in our lecture when we were talking about phospholipids, that was week 2, we took a look at the phospholipid and the fact that we had this hydrophilic head and we had this hydrophobic tail. And hydrophilic means that it loves water, and hydrophobic means that it hates water. And so because of this polarity of the phospholipid, when you put them in water, you drop a bunch of phospholipids in water, basically almost like click chemistry, they're going to align such that the hydrophilic heads are going to be close to the water, and the hydrophobic tails are going to be pointed away from water. And so you end up with this double layer. It's called a bilayer. Bi meaning two, right? You end up with this double layer of phospholipids, which is the plasma membrane. So if I wanted to create a synthetic plasma membrane, all I had to do is just take a bunch of phospholipids and put them into water, and they are going to make all of these plasma membranes. And so a lot of this knowledge, and I want you to remember, every time we learn something new in science, we try to determine how can we use that knowledge to better mankind? How can we use that knowledge that we just learned to create something that hopefully will help mankind, right? And so we are now in an age where we're trying to synthetically construct new organisms to do different jobs, right? So, say we had a spill in the ocean and an oil spill, and we wanted to actually create an organism that could eat the oil, right? Consume the oil, just like we have, the organisms down at the bottom of the ocean near these volcanic vents where they actually consume sulfur as their food, that perhaps we could create an organism that would consume the oil as its food and have a different byproduct so that we can remove the oil from the ocean. So we're using the knowledge that we can create synthetic organisms in different ways. And so the knowledge that in order to create a plasma membrane, all we have to do is drop a bunch of phospholipids into water and we get a plasma membrane. And this is exactly how every cell in our body, actually our plasma membranes are formed. Okay, so these phospholipids, really important. So every cell is bordered by a plasma membrane. It's surrounded by a plasma membrane. And these plasma membranes are made up of two layers, the bilayer, that are filled with a variety of pores. So this membrane is not just this monolithic bilayer. It actually has a lot of different structures inside of it. So, and you're going to learn more about what structures are there, but the functions of a plasma membrane are, one, to hold the contents of the cell in place, so it gives it some structure. The plasma membrane helps the cell to take in food and nutrition, and it can be selective about what it allows in and what it sends out. It aids in building and exporting different molecules. It allows interaction with the environment and also neighboring cells. We're going to see how that happens. So the plasma membrane is actually the gatekeeper of the cell. Okay. So we just talked about that. This is what I want you to remember. Why are plasma membranes such complex structures? Because they perform several critical functions: take in food and nutrients, dispose of waste products, build and export molecules, regulate heat exchange, regulate flow of materials in and out of the cell. The plasma membrane is extremely dynamic. But I want to take a closer look at the plasma membrane, because remember, we want to always get down to the molecular level, understanding how things work down to the molecular level. And here, what we can see on this slide, and I'm going to zoom in just a bit, we can see that we have these molecules that are embedded within the plasma membrane that help it perform these critical functions. So let me zoom in just a little bit more so you can actually hopefully see better. So here we have the plasma membrane in the sort of bluish color here. We have the hydrophilic region, remember the heads that love water. And then in this greenish sort of color, we have the hydrophobic region. These are the tails that hate water. And we can see the proper alignment based on its polarity. But we have these different carbohydrates that are there. We have these transmembrane proteins, trans meaning going across, that are actually crossing the membrane. These are very important. We'll talk about these transmembrane proteins. We have lipids inside of this phospholipid bilayer that makes up the plasma membrane. We have these different surface proteins that are present as well. And we're going to learn more about what each of these different components of these molecules, what they actually do. So let's shrink down there. So your take home is proteins found in the plasma membrane enable it to carry out most of its gatekeeping functions. So these proteins are extremely important. These proteins, these carbohydrates, they act as receptors and they help molecules enter and leave the cell, and they catalyze different reactions both on the inner and outer surface of the cell. So the question is, why do we even care about cell membranes and their function? Like, why do you even care? Why are you being taught this, cell membranes, right? It seems almost like it's just minutia. Like, okay, why do I need to know that detail? Well, it turns out that when cell membranes are actually faulty, then it can cause disease. So here we have the disease, which is called cystic fibrosis, and this occurs when an individual inherits from both parents incorrect genetic instructions for producing one type of transmembrane protein that allows chloride ions to get into and out of the cell. So one transmembrane protein that is the gatekeeper for chloride is faulty, it's defective, right? Now, this transport membrane occurs primarily in the membranes of cells in the lungs and the digestive tract. And so you would think, big deal, why that transmembrane protein doesn't work? Why do I care? Well, it turns out that when you are not moving chloride ions properly, because of this defective transmembrane protein, you get this buildup in the lungs of this thick, sticky mucus that gets produced. In someone with cystic fibrosis, this mucus collects in the lungs, impairing their lung function. They can't breathe, right? Increasing the risk of bacterial infection. And so they have to have something called coupage. And that's what you're seeing here in the left image. It's the image that's on my left. Might be on your right. I'm going to ping it so you know where I am. And so here we have someone with cystic fibrosis. They're laying down, and the person who is the caretaker, basically they cup their hands, hence the name coupage, and they pound their hands on the individual's chest, trying to loosen up the mucus so that they can cough it out and they can breathe again, right? And now, through technology, you can see here in the other image, we have this vest that helps by inflating and deflating rapidly, simulating that coupage, so that they can have this similar effect in the course of a 20-minute session by wearing this vest and moving the mucus out. So this defective transmembrane protein causes this whole disease. That's extremely important, right? But also, let's talk more about why we care about cell membranes, right? So let's talk about beta blockers. So question is, why do beta blockers reduce anxiety? So if you have anxiety, you may actually be prescribed beta blockers. So this knowledge of how cell membranes work allows us to make different drugs to control different conditions that we may have. So it's important knowledge, right? So drugs can alter cell membrane function, causing cell membrane dysfunction, but for good. Let's take a look at that. So it turns out that when you are under stress, what that actually is you have a small gland that sits right above the kidney called the adrenal gland. And the adrenal gland will pump out adrenaline, also called epinephrine. And this has an effect. So let's blow up the slide so you can see a little closer. Let's look at what's happening on the cell membrane. So here we have adrenaline that has been produced by the adrenal gland because the person is stressed. And when adrenaline locks into the receptor that's in the cell membrane, then this can actually cause a faster heartbeat, So when adrenaline locks into the beta receptor on heart muscle cells, then the heart muscle cell will beat faster, right? So the heart beat goes up. Blood pressure increases. So when the adrenaline locks into the beta receptor that's present on the vessels, it will cause the vessels to constrict, and that causes the blood pressure to go up. And so, we don't want that, right? That's a huge effect for someone who's, say, having an anxiety attack, or is having an issue with this production of adrenaline that's not wanted. Well, drugs have been made called beta blockers, hence the name. They actually block and they sit into the beta receptor, and in that way, adrenaline cannot sit into that receptor in order to initiate all those downstream effects. And so that's how beta blockers work. And so when you start reading, as you learn more and you go through this course and you start reading the package insert of different drugs and medications, and you start to see these terms, now you will start to understand what they mean when they say, oh, this is a beta blocker. You instantly will know, oh, now I know exactly how it works. It's actually blocking that beta receptor so that the target protein for that receptor cannot fit into it and cause the downstream effect of increased heart rate, increased blood pressure. Okay, so we can use that knowledge. Zoom back out. We can use that knowledge that we have of how these transmembrane proteins work for good. We can either stimulate a receptor, right, or we can block a receptor. So, take-home message. Normal cell functioning can be disrupted when cell membranes, particularly the proteins embedded within the cell membranes, do not function properly. However, we can also use this mechanism that we now know about as a positive and create different treatments, such as beta blockers, different receptor blockers. Okay. So now let's carry on with what's in a cell membrane. So cell membrane surfaces also have a, quote, fingerprint that identifies the cell. And so cells with an improper fingerprint are recognized as foreign. So your body basically knows this molecular fingerprint of you. There is a certain molecule, and we're going to see what that is, that basically is the molecule that's on every cell that you have that identifies you as you, and that is your molecular fingerprint. And so cells with an improper fingerprint can be recognized as foreign and attacked by your body's defenses. Well, that's bad, because that is a severe autoimmune condition, hence the name auto, self, immune. The immune system is attacking self, and that can be deadly. But this knowledge that we have, this molecular fingerprint on each and every cell, has helped us to understand why we get organ rejection in transplants. That's because, let's take a closer look at this image here. I'm going to zoom in again. That's because here we're looking at the donor liver, right? And it has the molecular fingerprint of the person that it came from, the donor. And here we have the actual person who's going to receive the donor liver, the recipient. They have their own molecular fingerprint, right, on each and every cell surface that they have. And so when you put this donor liver with this molecular fingerprint into someone, and it is not a match, it is a different look, you have one of two possible outcomes. Number one, the recipient's body can completely reject the donor liver, and the immune system will attack it and just destroy every cell, right? It recognizes it as foreign material. It does not have the self recognition molecules on it, right? So it will attack it. Or you can have the liver accepted after you give drugs to suppress the immune system so that it does not attack the new liver. And that's generally what we do because it's very, very rare to be able to find a perfect match for a donor to a recipient. And you'll see why when I show you what the molecule looks like. So you can match some of the things, but you may not be able to match all of the things. And so we can put someone who needs to have an organ transplant on these immunosuppressive drugs in order to allow the body to not attack the new donor organ. And that's kind of this tightrope. So you're trying to suppress the immune system enough that the body doesn't attack the new organ that you just transplanted in, but not suppress the immune system so much that the person is now going to basically be subject to every cold, mold, virus, anything that we normally can withstand. But if you suppress the immune system too much, then, it'll just ignore everything and they're going to die of something else, right? So it's a really fine line trying to titrate and find that amount of immunosuppressive drug that works to keep the person from attacking the organ, but still allow them to have enough of a functional immune system that they are not going to die of something else, another pathogen, right? But the other thing that's not on this slide is another possibility that is extremely rare that can happen when we have this donor and recipient situation. So here we looked at where the recipient will actually attack the graft, right? So this is host versus graft, and the host wins, right? But we can have this really rare situation where it's graft versus host, where the donor liver, for example, basically the cells that are in the liver will communicate, or whatever is actually being transplanted will communicate with the immune system and say, no, you are very confused. I'm the self. These are called histocompatibility complexes. These histocompatibility complexes that you see on me, these are the self. It's those other ones that are the foreign invader. And the immune system goes, oh, and starts attacking the recipient. And that is deadly, right? Because now the body is turning on itself. And that is one of the most deadly versions of a consequence that you can get with an organ transplant. Okay? So, because normally when you have host versus graft, you can do some biochemical measurements. You can see that the body is attacking, say, the donor liver. Things are not going well. It's not accepting the graft or the donor kidney. The body's not accepting it. The drugs that you're using to actually, take the immune system down are not working. And you have to remove the donor organ. Okay, you remove it. They go back on dialysis. And you wait for another organ to try to transplant. So that can be overcome, right? But when you have that super rare situation where it's graft versus host, that is very hard to turn off because now the body's, attacking itself. And that is one of the most dangerous situations that we see in transplantation surgery, right? But here I want you to have a look at what these molecular fingerprints kind of look like in sort of this graphical form, and you can see why it's so complex. So the molecular fingerprints that are on each and every one of our cells that I've been talking about, they're called the major histocompatibility complex. They also can go by the term HLA. But here what you can see, and again it's color coded, but it's also shape coded, is we have maternal major histocompatibility complexes. I'm going to call them MHCs from here on. So here we have the maternal MHC, and we have different classes of MHCs, major histocompatibility complexes. So we have class 1 and class 2s. You can see different conformations of them. We have maternal class 1, which means you have gotten this molecular fingerprint from the gene from your mother. And then we have paternal with a P, paternal MCH class 1 and class 2, which means you got these molecular fingerprints from your father. And so your cells contain a combination of both these maternal and paternal mean histocompatibility complex 1, class 1 and class 2. And now you can see why we have not been able to remove these complexes from the donor cells, because the next logical thing that you would say is, well, why can't we just, take these fingerprints off the liver or the kidney that we want to donate, and then that way we can just donate them into the recipient? And that's because it is extremely complex. And we have yet to find a way to remove all of these major histocompatibility complexes from each and every cell of an organ that we want to transplant. And the hope is that at some point, as the science matures, that we can use maybe CRISPR to go in and remove these major histocompatibility complexes and really change the whole landscape of transplantation, right? But CRISPR also allows sort of a larger vision than that, and we're going to talk about CRISPR when we get into DNA. But it also allows a vision of, potentially you won't need to actually have a donor organ. You can use your own DNA and through CRISPR change it and modify it so that now you're going to, in the lab, grow your own new kidney or grow your own new liver. And it's going to have all of your major histocompatibility complexes. It is you. And in that way, the transplantation is easy. It's you into you. So there's a lot that is coming as we learn more and more, right? But let's carry on with the cell membrane and disease, and why we need to understand how the cell membrane functions, because this is our basis of different diseases. So when we talk about HIV, here's the question: why is it extremely unlikely that a person will catch HIV from casual contact, such as shaking hands with an infected individual? Why does that actually not transmit the virus? Well, here's why. Because here on the cell membrane, we have these CD4 markers, okay? And these CD4 markers are only on cells that are within certain regions of the body. So they're not on our skin cells, right? So cells within the human body have CD4 markers, and HIV infects the body by binding to the CD4 marker on these cells. So here we have our plasma membrane, we have the CD4 marker that are only on certain cells, and then that is what the HIV virus is actually using to gain entrance into the cell. However, skin cells do not have CD4 markers, right? So skin cells on the surface of the body, because they don't have CD4 markers, HIV can't bind to these cells and they can't infect the body. And that's why you cannot catch HIV by shaking someone's hand. There are no CD4 markers to allow it to gain entrance. Okay. So the take home. Every cell in your body has a fingerprint. That's the major histocompatibility complex. And it's made of this variety of molecules on the outside facing surface of the cell membrane. This molecular fingerprint is the key to the function of your immune system, to recognizing you. Okay. I love this picture because this is basically a sheet of cells. Here we can see this is the cytoplasm with the different organelles. Here we've got the nucleus in blue with a nice DAPI stain on it. It's a type of stain that allows you to visualize the nucleus. Really nice. But let's talk transport. So, two types of transport that we want to know about. So we're going to start with passive transport first. And there are two types of passive transport that you need to know. One is diffusion and one is osmosis. So both diffusion and osmosis are types of passive transport. Passive meaning it doesn't take any energy for this motion to happen. Well, we're going to see exactly what that means in just a minute. But here's how to think of it very simply. When I'm talking diffusion, I'm talking about the movement of particles. When I'm talking about osmosis, I'm talking about the movement of water. Okay? That's how you can keep the difference between diffusion and osmosis clear in your head. Diffusion, movement of particles. Osmosis, the movement of water. Okay? So now let's look in depth at what each of these means. So, diffusion. Terminology. A solute is the particle. It's just another name for the particle, right? Doesn't matter if it's salt, it doesn't matter if it's particle of dye or ink, it's a particle, and it's called a solute. A solvent is the liquid or the gas that the particle is going to diffuse into or out of, okay? So here's how diffusion works. The way I like to think about it is particles like to get away from each other. Think of it as particles are just antisocial. They don't want to be with other particles. And so here is a nice image of, here I've got this the solvent, this liquid, say it's just water, and I put a drop of food coloring, and that food coloring has the particles of dye, right? So I can see it. And when I put that drop of food coloring into the water, what's going to happen? We're going to see the dye diffuse throughout the water, which means that the particles are spreading out. They're trying to get away from each other, right? Okay, so in diffusion, particles want to get away from each other. They don't want to be in a high concentration. They want to go where their concentration is lower. And so we call that a concentration gradient, okay? So they go from a high concentration gradient to a low concentration gradient. They just want to get away from each other. That is diffusion, okay? So how does that work in the body? Let's go ahead and kind of zoom in on this simple diffusion in the body. So we know that passive transport occurs when molecules move across a membrane without energy, right? That's what makes it passive. And so these molecules are going to move down their concentration gradient. They're going to go from a place of high concentration to a place of low concentration. It takes no energy for that to happen. That's just simple diffusion, just like dropping that dye into the water. They just spread, they get away from each other, right? They move to a lower concentration. So that's simple diffusion. And so there's different molecules that in our body that our body takes advantage of that fact, that knowledge, right? And so here we can see molecules of oxygen diffusing across the membrane of lung cells into the actual cells, while carbon dioxide diffuses in the opposite direction. So it's based on the concentration gradient. So here I have a high concentration of oxygen. Let me ping my cursor so you can see that better. High concentration of oxygen on this side of the membrane. It's going to, just through simple diffusion, want to come through the membrane into a area of lower concentration. And at the same time, I've got this high concentration of CO2 on this side, and it's going to want to diffuse in the opposite direction to where there's a lower concentration of CO2. That's just simple diffusion, takes absolutely no energy. It's just molecules moving across their concentration gradients from a high concentration to a lower concentration because they want to get away from each other, okay? But now we have something called facilitated diffusion, and now we're going to use some of these transmembrane proteins. Here we've got this channel or this carrier molecule, this transmembrane protein, and say the cell needs to actually move a certain molecule. So molecules can move across the plasma membrane with the help of a channel or carrier molecule. Okay, so here we've got the extracellular fluid, and basically this carrier molecule is opening up and saying, hey, you can come through. So it's acting like a gatekeeper, okay? So let's go ahead and shrink down once again so you can see the full slide. So in facilitated diffusion, most molecules can't actually get through the plasma membrane on their own. They want to get to the other side, right? Because the concentration gradient is lower, but they can't, maybe due to size, can't get through the plasma membrane. And so you can have this facilitated diffusion where you have these carrier molecules or transport proteins that will open up and allow them to come through. And so it's a way of gatekeeping. But it doesn't actually require any energy. All right. So defects in transport proteins can cause disease, right? That's why we care about these transport proteins. So defects can reduce or even bring facilitated diffusion to a complete stop. So just think that transport protein that's supposed to move this one molecule across the membrane is defective. It's not working. And so now that molecule either can't get in or can't get out, depending on what needs to be done. And this can have really serious health consequences. So for example, cystinuria, you have individuals where they basically are supposed to be moving cysteine out and they can't. And so it builds up and then that ends up forming these kidney stones because they can't move the cysteine out of the kidney cells, okay? So take-home message, for proper functioning, cells must acquire food molecules and other necessary materials from outside the cell, right? They have to sustain themselves, right? They have to get energy. Similarly, metabolic waste molecules and molecules produced for use elsewhere in the body have to move out of the cell. So when you think about it, we are this giant functioning mass of cells, right? They are organized into different organs and tissues and structure, but it all boils down to what these individual cells are actually doing. And the fact that we have waste to move out is because all of our cells are eliminating their waste, and we collect it into our bladder and we eliminate it, or we collect it into our colon and we eliminate it, right? All of these cells have to take in oxygen in order to create energy. We're going to see how that happens. And because of that, that's why we breathe in oxygen, because these cells need it. We just saw that oxygen actually gets diffused across the lung tissue just based on the concentration gradient, right? If the concentration of oxygen in my bloodstream right there at that interface of the lung tissue is low, then the oxygen that's on the outside is going to come in and cross into the bloodstream. If the concentration of CO2, which all my cells have excreted because they were creating their energy, and we're going to see how that happens on the molecular level, but they release their CO2 into the bloodstream, and now my bloodstream has a higher level of CO2. When it hits that interface between the outside air and the lung tissue in the bloodstream, and we're going to look closer at that, it's simple diffusion. More CO2 in the bloodstream, less outside, it's going to go outside. Okay? And that's going to become important, that knowledge of that, how these molecules are moving and how we're breathing based on simple diffusion really comes into play when we talk about going to high altitudes. We'll see how that actually, causes us an issue, why we have a limit, a ceiling as to how high we can go on the planet before we can't actually breathe anymore, right? Or why when we go diving and we have that scuba tank on, which has nitrogen in the tank, why we can get the bends, because the nitrogen is actually now forming into bubbles. So all of this knowledge that you have is really important as to why some of these other processes that you've seen happen, but you just didn't know why. Now you're learning more about why, okay? And also why, when you think about it, if you have someone, and this is why it's so dangerous, when you have people who live in very cold climates, maybe they've never been there before, and they turn on this this furnace that's not built to be inside of a house, it's built to be vented out, and you're getting this buildup of CO2 that's happening. When that concentration of CO2 gets higher inside the room, right, than it is in the bloodstream, where's the CO2 going to go? It's going to go into the bloodstream, right? And we're trying to get rid of the CO2. And so that's how people die, right? By not having proper venting when they're using different combustibles and different materials to try to stay warm. So knowledge of that is extremely important. All right. So in passive transport, which includes simple and facilitated diffusion and osmosis, the molecular movement occurs spontaneously without the input of energy. And this generally takes place as molecules are moving down their concentration gradient. So we talked about diffusion, but now let's look at osmosis. Now, when I talk about diffusion, we're talking about the movement of particles, but when I talk about osmosis, I'm talking about the movement of water. So osmosis is the passive diffusion or movement of water across a membrane, right? So there's some terms that you need to know, and I'm going to zoom in so you can see the slide on a closer view to get an understanding. All right. So here's the way I like to think of how water moves, and hopefully this will help you to remember how water moves. Water always wants to go where the party is. So where there is the greatest number of particles, that's where water will flow. Okay? So let's take a look. Here we have a cell. We've got two cells. We've got a plant cell and we've got an animal cell. Here, a red blood cell. Let's just look at the red blood cell just for simplicity. And we've got this red blood cell, and it has a certain number of particles that are inside of the cell, right, that are in the cytoplasm. And it's in this solution that has a certain number of particles. Maybe it's a saline solution, right? And if the number of particles that are in the solution are the same or equal to the number of particles that are in the cell, We call that an isotonic solution. Iso meaning the same. And so this is a balanced situation. Water's not going to move because there's no party going on. Everybody's the same. It's the same number of particles inside the cell as there are outside the cell. Water will stay where it is. However, if I have a hypotonic solution, now remember to lean on your Greek and your Latin. Hypo means below, it means less than, right? If I have a hypotonic solution, which means there's less particles in the solution than there are in the cell, there's more particles in the cell, water wants to go where the party is, water's going to go inside the cell, and that cell is going to swell up until it pops. Okay? That's a hypotonic solution. So less particles in the solution than are in the cell. Water's going to go where the particles are. It's going to rush into the cell, and they're going to explode. Okay? We can also have the situation where we have a hypertonic solution. Remember, hyper means above. Hyper means more. Someone who's hyperactive, they're extremely active, right? So hyper means more. So if I have a hypertonic solution, that means I have more particles in the solution than I have in the cell. Which way is water going to go? Water wants to go where the party is. It's going to go into the solution where all the particles are, and that cell is going to shrink down and shrivel. Okay? And so this is really important in the medical field because we want to make sure that we are giving the right type of solution for the problem that we're trying to solve. Okay? So most of the time, we are reaching for an isotonic solution because we're not trying to move water from different compartments at all, right? We're just trying to actually rehydrate the person, get more water into the bloodstream so that the tissues can actually go ahead and rehydrate. So usually we'll use an isotonic solution, right? But there are times where you may need to use a hypotonic solution, a solution where water is going to go into the cells, right? And so we'll do that if we have someone who, say, is extremely, let's say, dehydrated. We're trying to really get the fluids in fast. We can only use a little bit of these hypotonic and hypertonic solutions because we'll get this compartment expansion, right? And you can overload the vascular system because water is going to move based on the tonicity of the solution that you choose, right? So if I choose a hypotonic solution, then all the fluid that I'm putting into the body is going to go into the cells. And the cells are going to rehydrate, and if I'm not careful, I'm going to rupture all the cells, and I'm going to kill that individual, right? So that's why you can only use a little bit of a hypotonic solution and then kind of get this rehydration and follow it with an isotonic solution so that, water stays where it is. Same thing with a hypertonic solution. If I have a body that is extremely edematous, there's fluid in all the tissues, right? All the cells are just jam-packed with fluid. You could use a small amount of a hypertonic solution because where is the fluid going to go? It's going to go out of the cells and it's going to go into the actual solution itself. And the person's going to pee it out, right? I can give too much of a hypertonic solution and I'm going to basically shrivel all of their cells, pull all of the fluid out of their cells, and again, I just killed that individual, right? So when we talk about these using these extremes of solution tonicities, you have to be careful and you have to understand the tonicity of the solution that you're using, right? So I want you to make sure that you understand what isotonic solution means, and lean to your Greek and Latin to remember what it is, because it makes it simple. Iso is the same. It's a solution where the particles are the same as the cell that I'm talking about. Remember what hypotonic solution is. Hypo means less. That means there's less particles in the solution. Hypertonic solution means that there's more particles in the hypertonic solution. And then to remember which way water is going to go, water goes where the party is. It's always going to go where the majority of the particles are, okay? And that's osmosis, the movement of water. So when someone says the tonicity of something, they're talking about the relative concentration of the solutes, the particles, outside of the cell relative to the inside of the cell. So you can talk about the tonicity of the cell, you can talk about the tonicity of the solution, right? So it can be hypertonic, there's more particles. It could be hypotonic, there's less particles. It could be isotonic, it's the same. You have to follow it with the descriptor of what you're talking about. So I'm talking about an isotonic solution, right? Or I'm talking about the environment in the cell is isotonic, right? Or I'm talking about a hypotonic solution. Or I can say the environment in the cell is hypotonic, right? So you have to follow it with that description so the person knows which one of these are you talking about, right? All right. So how can we use what you just learned, this knowledge? All right, so say you've got, let me zoom in on this slide a little bit so you can see better. Say you've got a piece of celery. You just pulled it out of the refrigerator. It's all floppy, right? It's all dehydrated, it's flopping over. But you want to actually have a crispy piece of celery. How can we move water into that celery so that it's no longer dehydrated and we get a crispy piece of celery? So here we go. The dehydrated celery is going to contain these particles dissolved in water, these solutes, particles, relative to the water molecules, right? So if I were to place my piece of celery into distilled water, which means that the water it's distilled, which means not all of them, but the majority of the particles have been removed, right? Now I'm going to have more particles in the celery than I have in my distilled water. Where's the water going to go? The water's going to go where the party is, where the particles are. It's going to go into the celery, right? And the celery becomes crisp. There you go. Just solve the problem. But if I were to place my celery into salt water, let's think about it for a minute. So here I've got salt water. I've got all these salt particles in there, many more salt molecules and particles than I have inside the celery. Where's the water going to go? It's going to go where the party is. It's going to flow into the salt water, and my celery is going to continue to fade, right? The celery is going to become even more shriveled because the water is going to leave the celery, okay? So we can use the knowledge that we have. So that's osmosis in action. But probably at some point, someone that you know has had to use a laxative, right? Things have gotten a little bound up in the GI tract. You can't actually have a bowel movement, and the doctor may direct you to either, take some milk of magnesia or some magnesium salts. Like, why does that make you poop, right? Why is that going to solve the problem? Well, think about it. So milk of magnesia, which means we've got all these magnesium particles, or magnesium salts, which means we've got all of these magnesium salt particles that are in there. When you take that in, you've got a high particle concentration relative to what's going on in your intestine, and the water is going to move via osmosis from the cells into the intestine. Suddenly you've got all this water in the intestinal tract, and whoosh, you just flush things out, right? And someone a long time ago made a lot of money off of that knowledge when they created milk of magnesia, just based on the knowledge of osmosis. Because it turns out that water doesn't care what the solute is. Water doesn't care what the particle is. As long as there's more particles, that's where the water is going to go. It doesn't matter what those particles are, okay? So it's only determined, the direction of osmosis is only determined by a difference in total concentration of all the molecules dissolved in the water, and it doesn't matter what those particles are, okay? All right. Let's carry on. So let's continue our exploration of the cell because we're getting there. Active transport. So we talked about passive transport. That's our osmosis and our diffusion, but there is something called active transport. And active transport requires ATP. So cells use energy to move small molecules into and out of the cell. Molecules can't always move spontaneously and effortlessly into and out of the cell. They actually need some form of energy or transport in order to cross the cell membrane. You can't do it through osmosis or, diffusion, okay? So there are two distinct types of active transport. There's primary and secondary, and they differ only in the source of fuel. I only want you to remember primary transport, okay? So primary active transport uses energy directly from ATP. I have been known to put this image, which I am going to blow up here so you can see better. Sorry. Right there. This image onto an exam. And the question is, what type of transport is this? And you can see these molecules of ATP, and I'm going to try to blow it up a little bit more and not have it leave the screen on you. Right there. Sorry about that. These molecules of ATP. When you see ATP in transport, it's active transport. Done. Easy, simple. I see the use of ATP. It's active transport. It turns out that our bodies actually use active transport to get hydrogen ions into our stomach. That's how you get your stomach acid. So let's shrink down a little bit more so you can see more of the bigger picture here. So active transport occurs when the movement of molecules into and out of a cell requires the input of energy. For example, in response to eating, the cells lining your stomach use ATP to pump large numbers of hydrogen ions into the stomach. Remember, more hydrogen ions, that makes acid. Remember our pH lecture? So here we have inside a cell that's lining the stomach, right? And we here we have the outside of the cell, which is the inside of the stomach, okay? So here we can see we've got this high concentration of hydrogen ions inside the stomach. But the cell needs to get more hydrogen ions into the stomach. Well, the hydrogen ions aren't going to want to go in there through diffusion because it's a high concentration and particles don't like each other. They want to stay apart, right? So they're not going to move by diffusion. And so what has to happen is what's called active transport, where the body is actually pumping them into the stomach. And that requires ATP, okay? So when you see ATP being used to transport, you know that it's active transport, okay? I'll just briefly touch on secondary active transport. In secondary active transport, no ATP is used directly, but it's used at some other point in some other location. For example, you pump some water up to the top of a tower. You used energy to pump it up to the tower. Now it's sitting there, and at some future time, you can actually open up the spigot and the water will run down. Okay? So when you open the spigot and the water runs down, you're not using any energy at that point, but you did have to put energy into the system to get it up to the top. It just wasn't at that same moment in time. That's secondary active transport. Okay? I don't want you to get too caught up on it. I just want you to remember primary active transport. When you see the use of ATP, you know it is primary active transport. Okay? So we will move on. In active transport, moving molecules across a membrane requires energy. This is sort of a recap. And active transport is necessary if the molecules to be moved are very large, or if they're being moved against their concentration gradient. They're being moved in a direction that they don't want to go. Then active transport can be used to actually move them. But there's another way that a cell can actually get particles inside that it needs. So terminology here. It can use what's called endocytosis. Again, I want you to lean on your Greek and your Latin prefixes and suffixes. So endo means inside. Cytosis is the cell. So endocytosis, inside the cell. Exocytosis, exo means outside, cytosis the cell. So exocytosis is when the cell is putting something outside the cell. Okay? So a cell can either bring particles inside that are just too big to get inside through passive or active transport. They won't fit into those transport channels. It's too large a molecule. Or if it needs to excrete a molecule or a particle that's too big, it has a mechanism to excrete it. So let's look at what that looks like. So here's an image of an actual cell engulfing another cell, and engulfing that cell, we call that phagocytosis. Phago to eat a cell eating, right? But phagocytosis is a type of endocytosis. It's taking something into the cell, okay? So that's what that looks like. So there are 3 types of endocytosis. One is phagocytosis, which we just saw, cell eating. Pinocytosis, which is cell drinking. And the way I remember pinocytosis is Pinot Grigio. So pinocytosis is cell drinking. And then we have receptor-mediated endocytosis. And this type of endocytosis is really important because this is what the COVID-19 virus uses to gain entry to our cells. So we're going to see what that looks like, but it's a type of endocytosis, okay? So 3 types of endocytosis, bringing things into the cell. One is phagocytosis, cell eating. The other is pinocytosis, cell drinking, bringing in a liquid. And the other is receptor-mediated endocytosis. All right, so let's take another visual look at those a little bit closer. So here we have phagocytosis that you saw before, where the cell is engulfing something very large. It's eating something that it wants to obtain that's quite large, okay? So how does this happen? Step 1, the plasma membrane forms this pocket-like vesicle around the large particle. Step 2, the particle is transported into the cell inside this vesicle. So when it engulfs it, when it phagocytoses or phagocytizes a particle, it's putting it in a vesicle, okay? It's putting it in its own little storage unit. That's what a vesicle is. and it will deal with it depending on what that particle is, okay? So, pinocytosis is the process of cells taking in dissolved particles and liquid. Okay, so same thing. It's going to put it into a vesicle. Receptor-mediated endocytosis. Now, I want you to take a very close look at that, and let's make sure you understand this, because this is the mechanism that COVID-19 co-opts in order to get into our cells. So here we're going to look at this process, this receptor-mediated endocytosis, this process looking at a cholesterol, right? A piece of cholesterol. So we have a cholesterol protein here, and we've got this LDL particle and a liver cell. Okay. So step one, the LDL receptor protein, that's this little knob here. Let me see if I can zoom in so you can get a much closer look. The LDL receptor that we see here, the receptor proteins are on the plasma membrane of the liver cells, and they specifically recognize and bind to LDL. So we have the protein itself, and then we have the receptor. And here the receptor, the LDL protein receptor, which is going to sort of match, like lock and key to the actual protein. It's very, very specific, the protein that it recognizes. So when the receptor recognizes the protein and it locks on, then it will begin to pull in the particle that contains the recognition proteins, right? And it's going to put it into a vesicle. And we'll see later when we dive deeper into COVID-19 how it co-opts this, but it has its own little proteins called the spike proteins, the S proteins, that actually fit perfectly with our ACE2 receptor. And we'll see that when we get to COVID-19. I have a visual, but it's the same thing, just different proteins and receptors. And when it locks on, then the cell brings it in. Okay, and that starts the whole process of COVID-19. But the virus itself is co-opting a process that we have naturally, that we use to bring in particles that our cells need, right? So in this case, the cell needs cholesterol, and it needs it to, make estrogen and testosterone. So it has very specific receptors for very specific things that the cells need. But lots of pathogens have learned, evolved, adapted to co-opt these, our receptor-mediated endocytosis. Now, exocytosis is sort of the opposite of that same path that we just saw. Let's sort of zoom in. So we have a particle that's inside of a transport vesicle. The cell needs to get rid of it. It needs to eliminate it. It has tagged it as waste. It will, step one, the molecules are packaged into the vesicle within the cell. The vesicle then fuses with the cell's plasma membrane, and then the vesicle contents are released for use throughout the body. So if it's waste, then it's going to go off to be processed in the liver or the kidney and be eliminated as waste. But it may be, say this is a cell that is in your adrenal gland, and what's inside of this transport vesicle is actually adrenaline. And so it will actually leave in this same way, and so adrenaline will actually be exported through exocytosis, and it'll go into the bloodstream to have those downstream effects that we saw before on the heart and the blood vessels, causing anxiety. But basically, it's not just causing anxiety, right? That's your fight or flight mechanism. So that is your body's response to something terrible has happened that I'm perceiving, right, through the eyes or some of your other senses. And so through the brain and the hypothalamus, which basically is communicating with the adrenal glands, because everything is interconnected, which is what we have to remember, right? Something is causing that response that's causing a release of chemicals, hormones, cytokines, that basically are communicating with the adrenal gland, requesting that adrenaline be released so that the heart rate can go up, so that we pump more blood, we get more glucose and oxygen to the cells because we need to run, right? Maybe you looked out and you spotted a lion, right? Suddenly you're going to start that whole cascade. You're going to get the release of adrenaline. Heart rate's going to go up. Blood is going to start pumping. The breathing, the respiratory system is going to increase because we're trying to now increase the oxygen that's happening, diffusing into the bloodstream so that it's available for the muscles so that we can run, fight or flight, okay? So you can see how all of this is now being tied together, all of these processes, all this knowledge that you have been given bit by bit, how it all interconnects, okay? To form some of the processes that you know very well, right? But let's carry on. So let's take a look at how cells are connected and how cells communicate. Let's see if we can get a nice size here. There we go. So connections between cells hold them in place, number one, but they also enable them to communicate with each other. And this involves numerous types of proteins and glycoprotein adhesion molecules. So glycoproteins, what are those? Glyco, meaning glucose, sugar, protein. It's a sugar and a protein put together, hence the name glycoprotein, okay? So communication is important between cells. So let's look at the three primary connections that we can have between animal cells. We are considered animals, and so these are the type of connections that our cells have. Number one, there's a type of connection between two cells called the tight junction, and this forms a watertight seal between two cells. It's kind of like caulking around a tub, right? Between those two cells, it's watertight. Nothing is getting between there, not even water. And tight junctions are important because those are the junctions that we actually have in our brain. And that's what protects our brain from having different waste products and chemicals get to the brain. It's because of these tight junctions. It also makes it really difficult for us to treat conditions of the brain because we cannot get very many of our drugs to actually cross what's known as the blood-brain barrier, which is the series of tight junctions, okay? So that's why knowledge of tight junctions is important. That is what is protecting our brain. But then we also have what's called desmosomes. And desmosomes kind of act like the Velcro and fastener that you're used to, where you just, pull them apart. These are connections that we have between, say, heart muscle cells, these little Velcro type connections. And they're keeping the cells together. But also we have what's called gap junctions. And these are really important. These are really cool. These are kind of like secret passageways that are in a house where you're just like, you move that bookcase and there's like another room behind there. Cells have these secret passageways called gap junctions where they can actually pass these chemical messages between each other, between cells. Okay, those are the gap junctions. It's really, really cool. So In multicellular organs, most cells are actually connected to other cells. They're not just sort of, out there hanging free. That's how we get our tissues. That's how we have our form and our function. All right. So now we're getting down almost to the end. We're almost there. So there are 9 important landmarks to distinguish eukaryotic cells. And I need you to know these different organelles and these different bits and pieces that make up eukaryotic cells. So number 1 is the nucleus. And the nucleus, so you're looking at this chart, and this chart basically has, which of these organelles are in plants, which are in animals. You already know that plants are the only ones that have cell walls. Animal cells do not have cell walls. Plants are the only ones that have chloroplasts. Animal cells do not have chloroplasts. That's the big difference, right? Those two. All right. So you need to know what the nucleus is and what its function is. The nucleus basically directs cellular activity and stores hereditary information. You have the cytoskeleton. So cells have their own form of skeleton and structure, right? It's not like bone, but it's these fibers that run throughout. It's like cabling that you'll see inside the cell, and it's called the cytoskeleton, and it provides structural shape and support and enables cellular movement. So cells can actually move if they choose to. The mitochondria, that is known as the powerhouse of the cell. We're going to talk more about that when we talk about energy. But right now, just remember that the mitochondria is the powerhouse. It harvests energy for cellular function. Remember, we talked about the mitochondria in terms of the endosymbiosis theory. Then we have something called the lysosome. The lysosome basically digests and recycles cellular waste products and consumed material. So the lysosome is really cool because it's this acid pit, essentially. It is a vesicle that is filled with acid. And so when a cell wants to break something down or destroy it, it will put it into a lysosome. So say it actually, through receptor-mediated endocytosis, it brings in a foreign particle, right? And it recognizes that particle as foreign, it will immediately put it into a lysosome and just acid bath destroy it, right, into its pieces. The rough endoplasmic reticulum, also known as the rough ER. So the rough ER is studded with ribosomes, which is why it's called the rough ER, because they look like little studs all over the rough endoplasmic reticulum. And the ribosomes are where we create our proteins, okay? So the rough endoplasmic reticulum modifies proteins that will be shipped elsewhere inside of the organism or the cell. So we have the rough endoplasmic reticulum. We also have the smooth endoplasmic reticulum. The smooth endoplasmic reticulum is where we synthesize our lipids and we detoxify some of the molecules that we need to detoxify. The Golgi apparatus. I like to think of the Golgi apparatus as the post office of the cell. After a cell makes a protein, it sends it to the Golgi apparatus, and the Golgi apparatus basically processes and packages that protein, lipid, steroid, whatever that molecule is, and it puts a molecular stamp as to where it is to go. Is it to be exocytosed outside of the cell, to go to another part of the body? Is it to go to another part inside the cell. So the Golgi apparatus is actually doing all of that packaging and processing of a protein or a molecule that the cell created. Okay, we have the cell wall, which is only in plants, right? And that provides structure and strength. We have a vacuole, which is kind of similar to a vesicle, but vacuoles are more like storage systems. So it's like the garage of the cell. And animal cells can sometimes have it. Plant cells use vacuoles a lot because they store a lot of different things. But here's where nutrients can be stored, degraded waste products, different types of pigments in plants, structural support, especially in plants. Vacuoles can lend some of that. So just think of a vacuole as a storage place, and both plants and animals can have it. Plants use them the most. And then of course we have chloroplasts, which is where photosynthesis is being performed, and that's only in plants. All right, so know this chart, know what these different organelles are. Let's carry on. The nucleus is the cell's genetic control center, right? The nucleus is the largest and most prominent organelle in most eukaryotic cells. Remember, prokaryotic cells don't have a nucleus. The nucleus has two primary functions: the genetic control center and the storehouse for hereditary information. When we look at the nucleus, I'm going to zoom in on this slide so you can see hopefully a little closer. Let's go through some terminology. So the nucleus functions as the genetic control center of the cell, stores hereditary information, but inside the nucleus is another area called the nucleolus. And this is where those ribosomal subunits are assembled. So remember I told you that the ribosomes are these little balls that are sort of studded on the rough endoplasmic reticulum. That's where proteins are made. Well, the nucleolus is where the ribosomes are actually made. Really cool, right? And then they're exported outside of the nucleus and they go to the rough endoplasmic reticulum. Now, inside the nucleus, you can see in sort of this blow-up picture, we've got chromatin / chromosomes. And these are thin fibers of DNA which carry all the hereditary information. The nucleus has these, it's got a nuclear membrane, of course, which is a bilayer, but it has these nuclear pores. And this is how RNA can come into and go out of the nucleus. And we'll see why that's important. So the nucleus has a nuclear membrane. It is your standard, phospholipid bilayer, as you can see here, but it has nuclear pores so that we can get RNA into and out of the nucleus, because DNA cannot leave the nucleus. It's trapped there. It does not leave the nucleus, but we've got to get that information out, and we'll see later how we get that information out when DNA can't leave. Let's carry on with our terminology. So chromatin. Chromatin is a mass of long, thin fibers consisting of DNA with some proteins attached. Okay. Nucleolus. It's an area near the center of the nucleus where subunits of the ribosomes are assembled, and the ribosomes are our little protein factories, and they make their way out into the rough endoplasmic reticulum. This is just giving you a closer picture at the cytoskeleton, and its three chief purposes. So it's made up of these microtubules, and it has these intermediate filaments and these microfilaments. So it has structure. And the cytoskeleton is giving the cell structure. It is not made of bone, by the word skeleton, that's immediately what you would think of. But this is the cell skeleton, and it's made up of microtubules and microfilaments. Okay? So the cytoplasm, excuse me, the cytoplasm and the cytoskeleton form the cell's internal environment, and they provide the cell's physical supports and aid the cell in movement if the cell needs to move. Now, more parts of a cell. Now, not all cells have cilia and flagellum. Cilia are these small hair-like projections, and they're inside the lining of our lungs. And when you think about it, when we're breathing in, there's lots of particles in the air, right? There's lots of little dust particles and like dander and things like that in the air. We're not breathing like ultra filtered air, right? It's just air, and it's got lots of particles in it. And those are coming into the lungs. Well, how do you get rid of that? You get rid of that by use of these cilia. And so they're always sweeping in the outward motion, right? They're sweeping all of the air that's coming through, and they're collecting these particles, and they're moving them up until they get enough, and that elicits a cough. And you clear what is in your lungs. So coughing is very important to clear your lungs of all the particles that you have breathed in. That's why we all cough, right? We don't cough all the time, but we all cough because our cilia are active and they're doing their job of clearing out the lungs, okay? And that's really important. So not every cell has cilia. Only certain cells of the human body have cilia, such as in the lungs. Now, flagellum. It turns out that sperm are the only cells in the human body that have flagellum. No other cell in the human body has flagellum. And it is a tail-like feature that's used for locomotion, for movement. Okay? We're going through the different organelles just to make sure you understand all the organelles on that chart. And here is our friend, the mitochondria, which is one of the organelles that prompted the endosymbiosis theory. Do you remember what the other one is? It was the mitochondria and chloroplast. That's right. So mitochondria in both plants and animals, and also chloroplast in plants, prompted the endosymbiosis theory. Okay, so this is what the mitochondria looks like. We're going to talk about it much more when we get into energy, but it's basically what's called a double bag. You can see we've got an outside membrane here, and then we've got an inside membrane that is folded, hence the name double bag. And this is where the energy that we all use is created. So it's this bag within a bag structure. Okay, that is the mitochondria. And we already talked about the endosymbiosis theory to try to explain why mitochondria, which have their own DNA, exist inside of cells. So we have two places where we house DNA in our cells, right? We have DNA in our nucleus, and that's the traditional DNA that we think about that, brings about all of our phenotype, all these features that we see, comes from the genes that are encoded in our DNA that's in the nucleus. But we have a separate set of DNA in our mitochondria that specifically comes from our mother, and that presence of this organelle that has its own DNA is explained through the endosymbiosis theory. All right. So it turns out we have, if I ask you the question, we all have more DNA from one parent than the other. Who is the bigger contributor? Well, that's going to be your mom, because you've got, half your DNA that's in your nucleus from her, but you also have all the DNA that's in your mitochondria from her as well. All right. Carrying on, we are almost there. Form follows function in an organism's cells and reflects their environment. When cells must perform intensive heat production, they significantly increase the number and size of their mitochondria. So the number of mitochondria that we have in our cells is not static. We can actually increase the number of mitochondria that we have in our cells when we have a need for more energy over time. So, for example, people who are athletes will use this knowledge, their trainers will use this knowledge by training them to perform a function, right, over time. Now their muscles are needing more energy, right, all the time. What happens? The muscle cells are going to increase the number of mitochondria that are in the muscle cells, which is great because mitochondria make energy. And that way, when it's time to run that race, they have the extra mitochondria. They're ready to run that race, okay? There are other ways to increase mitochondria. We'll talk about that later. But the number of mitochondria that you have in your cells is not static. You can actually make more, and you do make more, depending on demand and need that you put on your body and the particular cells, okay? Lysosomes, we talked about that. When we looked at the chart, they are the cell's garbage disposal. They are the acid bath. When something needs to be destroyed or broken down, it is put into a acid bath lysosome. Super cool. Cell gets all medieval on it. All right. And lysosomes are filled with about 50 different digestive enzymes, which is really cool, in addition to the acids, and they enable a cell to completely dismantle macromolecules, including disease-causing bacteria. They will put a bacteria or a virus into a lysosome and completely destroy it. It's really cool. You would think we would have made a drug that would tag a virus to immediately go into a lysosome. Yeah. We haven't, but we should. All right. The endoplasmic reticulum. This is, there's two of them, right? There's the rough endoplasmic reticulum and there's the smooth endoplasmic reticulum. And so here we can see, I'm going to sort of zoom in so you can get a closer look here at the rough endoplasmic reticulum. We can see that we've got the ribosomes, which is where we make our proteins, studded on the rough endoplasmic reticulum, right? And then here we have the smooth endoplasmic reticulum, and this is where we're detoxifying a lot of the drugs and things that are brought into the cell. We're breaking down chemicals and toxins using the smooth endoplasmic reticulum. So the rough endoplasmic reticulum, we're making our proteins. The smooth endoplasmic reticulum, we're breaking down toxic chemicals, cellular byproducts. Okay. Let's go ahead and shrink down again. This is just another look at the rough endoplasmic reticulum, and you can see this is an actual electro micrograph of the rough endoplasmic reticulum, in addition to sort of the cartoon view that you'll see with it studded with ribosomes. Okay. This is another look at the smooth endoplasmic reticulum. Function: synthesize our lipids, such as our fatty acids, our phospholipids. So when we're talking about the phospholipids that make our cell membranes, if you're wondering, well, where do those come from? They're made right here in the smooth endoplasmic reticulum. So that's where we are taking our atoms that we have taken in from our food, and we broke them down into the individual atoms, right? And we bring them over here into our smooth endoplasmic reticulum, and we're basically creating those bonds to make our phospholipids. Okay, so this is where we're making our fatty acids, our phospholipids, our steroids, We're detoxifying molecules such as alcohol. So when you go to have that margarita and you're wondering, like, yeah, I know my liver detoxifies it, but that's kind of vague, right? The liver detoxifies it. Well, this is the portion of the cells in the liver that actually are detoxifying the alcohol, the smooth endoplasmic reticulum. Okay. Different drugs are broken down using the smooth endoplasmic reticulum. So it's super, super important. And you can imagine how things can go awry if that malfunctions. All right. So let's take a look at your take-home message. The Golgi apparatus. Do you remember what the Golgi does? It's the post office for the cell, right? The Golgi apparatus processes products for delivery throughout the body. It tags it as to where it's going to go. This is what the Golgi looks like. And you can see it both in its cartoon animation form. This is a transport vesicle sitting here. And what it actually looks like when you're looking at it inside a cell. This is, again, an electron micrograph. When you look through your standard microscope, you cannot see to this level of detail. That's how small it is inside the cell. But as you can imagine, super, super important, because things, if they are mistagged, then they will go to the wrong place, and that can cause disease and a problem. So our Golgi apparatus, the post office of the cell. If the cell is a plant, then we have the cell wall, and these you can easily see under a regular microscope. I'm just going to make this slide go down just a little bit. There we go. And so this is a view, and so this is how cell walls actually are interconnected, and that gives the strength. We use that knowledge, right? In plants, we use their cell walls, and we call it wood, and we build buildings with it. So a cell wall can confer a tremendous amount of strength. As you know, wood is strong, right? A tremendous amount of structural strength on plant cells, and it gives plants this increased water resistance, provides some protection from insects and other animals that might want to eat the plant. And in plants, plasmodesmata connect the cells and enable communication and transport between them. Here we are just going over the vacuoles that we already talked about. So this is multi-purpose storage sacks for cells where things are stored inside vacuoles. Both plants and animal cells have them. So in plants, they can have something called a central vacuole that you can easily see under a microscope. And the central vacuole can actually play an important role in 5 different areas of plant life. Number 1, nutrient storage, waste management, predator deterrence, meaning this is where a lot of the plants will store its toxins. When you're just like, that plant's toxic, don't eat it. Well, the toxins are going to be stored in these vacuoles. Sexual reproduction, the vacuoles come into play, and physical support. Okay, so in plants, the vacuoles are super, super important. Chloroplasts. This is what they look like. We're going to go deeper into chloroplasts when we get into energy, but they are the power plant for the cell so far as photosynthesis. This is what it looks like. This is a real image of it. This is sort of the animation cartoon image of it. Inside, they have what's called thackyloids, and we'll learn more about these thackyloids, but this is where photosynthesis is actually happening inside the chloroplast. And so we have both stroma, which essentially is sort of all of the inside material, if you will, and these interconnected little flattened sacs that basically act like these energy-creating batteries called the thackyloids. That's these little stacked battery-looking things. That's what's actually creating the energy through photosynthesis inside the chloroplast. All right. So now we've had a complete walkthrough through that entire chart. We've gone over that entire chart several times. So now I just need you to get yourself familiar with each of these organelles and make sure you understand what they are and what they do. And I want to show you a quick movie that gives you this really nice animated view inside the cells. Okay, so I want to just show you guys this movie of the inner workings of a cell, and I want you to see how dynamic the inside of a cell actually is. You just learned all the parts and the organelles that are in there, but I want you to see them in action because I think it really brings home how intricate the system is. And let's just take a look at it.
While red blood cells are carried away at high velocity by a strong blood flow, leukocytes roll slowly on endothelial cells. P-selectins on endothelial cells interact with PSGL1, a glycoprotein on leukocyte microvilli. Leukocytes, pushed by the blood flow, adhere and roll on endothelial cells because existing interactions are broken, while new ones are formed. These interactions are possible because the extended extracellular domains of both proteins emerge from the extracellular matrix, which covers the surface of both cell types. The outer leaflet of the lipid bilayer is enriched in sphingolipids and phosphatidylcholine. Sphingolipid-rich rafts raised above the rest of the leaflet recruit specific membrane proteins. Rafts' rigidity is caused by the tight packing of cholesterol molecules against the straight sphingolipids hydrocarbon chains. Outside the rafts, kinks in unsaturated hydrocarbon chains and lower cholesterol concentration result in increased fluidity. At sites of inflammation, secreted chemokines bound to heparan sulfate proteoglycan on endothelial cells are presented to leukocyte 7 transmembrane receptors. The binding stimulates leukocytes and triggers an intracellular cascade of signaling reactions. The inner leaflet of the bilayer has a very different composition than that of the outer leaflet. While some proteins traverse the membrane, others are either anchored into the inner leaflet by covalently attached fatty acid chains or are recruited through non-covalent interactions with membrane proteins. The membrane-bound protein complexes are critical for the transmission of signals across the plasma membrane. Beneath the lipid bilayer, spectrin tetramers arranged into a hexagonal network are anchored by membrane proteins. This network forms the membrane skeleton that contributes to membrane stability and membrane protein distribution. The cytoskeleton is comprised of networks of filamentous proteins that are responsible for the spatial organization of cytosolic components. Inside microvilli, actin filaments form tight parallel bundles, which are stabilized by cross-linking proteins. While deeper in the cytosol, the actin network adopts a gel-like structure, stabilized by a variety of actin-binding proteins. Filaments, capped at their minus ends by a protein complex, grow away from the plasma membrane by the addition of actin monomers to their plus end. The actin network is a very dynamic structure, with continuous directional polymerization and disassembly. Severing proteins induce kinks in the filament and lead to the formation of short fragments that rapidly depolymerize or give rise to new filaments. The cytoskeleton includes a network of microtubules created by the lateral association of protofilaments, formed by the polymerization of tubulin dimers. While the plus ends of some microtubules extend toward the plasma membrane, proteins stabilize the curved conformation of protofilaments from other microtubules, causing their rapid plus end depolymerization. Microtubules provide tracks along which membrane-bound vesicles travel to and from the plasma membrane. The directional movement of these cargo vesicles is due to a family of motor proteins linking vesicles and microtubules. Membrane-bound organelles like mitochondria are loosely trapped by the cytoskeleton. Mitochondria change shape continuously, and their orientation is partly dictated by their interaction with microtubules. All the microtubules originate from the centrosome, a discrete fibrous structure containing two orthogonal centrioles and located near the cell nucleus. Pores in the nuclear envelope allow the import of particles containing mRNA and proteins into the cytosol. Here, free ribosomes translate the mRNA molecules into proteins. Some of these proteins will reside in the cytosol. Others will associate with specialized cytosolic proteins and be imported into mitochondria or other organelles. The synthesis of cell-secreted and integral membrane proteins is initiated by free ribosomes, which then dock to protein translocators at the surface of the endoplasmic reticulum. Nascent proteins pass through an aqueous pore in the translocator. Cell-secreted proteins accumulate in the lumen of the endoplasmic reticulum, while integral membrane proteins become embedded in the endoplasmic reticulum membrane. Proteins are transported from the endoplasmic reticulum to the Golgi apparatus by vesicles traveling along the microtubules. Protein glycosylation, initiated in the endoplasmic reticulum, is completed inside the lumen of the Golgi apparatus. Fully glycosylated proteins are transported from the Golgi apparatus to the plasma membrane. When a vesicle fuses with the plasma membrane, proteins contained in the vesicle's lumen are secreted, and proteins embedded in the vesicle's membrane diffuse in the cell membrane. At sites of inflammation, chemokines secreted by endothelial cells bind to the extracellular domains of G-protein-coupled membrane receptors. This binding causes a conformational change in the cytosolic portion of the receptor and the consequent activation of the subunit of the G-protein. The activation of the G-protein subunit triggers a cascade of protein activation, which in turn leads to the activation and clustering of integrins inside lipid rafts. A dramatic conformational change occurs in the extracellular domain of the activated integrins. This now allows for their interaction with ICAM proteins displayed at the surface of the endothelial cells. These strong interactions immobilize the rolling leukocyte at the site of inflammation. Additional signaling events cause a profound reorganization of the cytoskeleton, resulting in the spreading of one edge of the leukocyte. The leading edge of the leukocyte inserts itself between endothelial cells, and the leukocyte migrates through the blood vessel wall into the inflamed tissue. Rolling, activation, adhesion, and transendothelial migration are the four steps of a process called leukocyte extravasation.
So I wanted you to see that, to see how incredible it is that all of these processes are going on inside each and every one of our cells throughout our body. And this movie puts it in the context of a white blood cell that is, rolling along the tissues and going to a site of inflammation. But what's important to this movie is that you don't try to take notes on this movie, that I just want you to sit back and watch and see the complexity that's happening inside each and every one of our cells. And even though we're talking about each of these parts, the ribosome and the nucleus and the Golgi apparatus and the mitochondria in these little discrete bits and pieces, it's important to kind of see how it's all interacting and it's all unfolding each and every day without us even thinking about it. That's what's so amazing.